Okay, welcome everyone. See more people streaming in. Welcome, welcome. All right, I guess we'll get right to it and we'll have more people probably streaming in as we go. I see a couple familiar names, so if you don't already know, my name is Rachel. I'm the Environmental Education Coordinator here at Reforest London. I currently work at the Reforest office located at the Westminster Pond Centre in London, Ontario, which is being developed as London's first Centre for Environment and Sustainability. This is our 7th webinar in our Solutions at Scale virtual education series. We're super excited to be running post-conference these spotlight sessions and to join in the discussion today with our speaker. We're situated on the traditional territory of the Attawandaron, Anishinaabeg, Haudenosaunee, and Lenape peoples, nations which have long-standing relationships with the land, water, and region of southwestern Ontario. We extend our recognition to our neighboring First Nation communities, the Chippewas of the Thames First Nation, Oneida Nation of the Thames, and the Munsee Delaware Nation, who continues to steward the land today. We recognize the treaties of this area, such as the London Township Treaty of 1796 and the Dish with One Spoon Covenant Wampum, among others, and understand that we are all treaty people. So we're super excited to be hosting Colleen Dempster today. Hi, Colleen. Welcome. Colleen is passionate about restoring the environment. In recent years, she turned her attention to rewilding soil and in turn boosting the health of plants and people. She obtained a master's in environmental and life science from Trent University, a postgraduate certificate in ecosystem restoration from Niagara College, is an ISA certified arborist, and is certified by Dr. Elaine Ingham's Soil Foodweb School as a laboratory technician. She has worked to help arborist companies develop compost tea programs and now works as an educator at Fleming College and as an independent consultant who makes high quality compost teas and extracts from her home in rural Uxbridge. So thank you so much, Colleen, for being here today. We're super excited for your presentation. Just before I hand it over to you, a couple housekeeping notes. We are going to be ending, as usual, this webinar with a Q&A. So think of your questions throughout, and we'll get to those at the end. Feel free to use the chat to introduce yourselves. Let us know where you're zooming in from. And if you haven't already, check out our Solutions at Scale page on the Westminster Ponds website to read more about upcoming sessions and previous sessions which have been recorded and are being posted to our YouTube. So yeah, thanks so much. Without further ado, I'll hand it over to you, Colleen.
All right, thank you so much, Rachel. And it's a pleasure to be here. I am loving my newfound career of educating people about restoring the environment with organic soils and building organic liquid amendments to help accelerate that restoration process. I've been doing this for about 6 years, and I just see more passion and more excitement and more evidence that it's working, and it's building a really positive community that overlaps with sustainable agriculture, regenerative agriculture, like I said, organic practices, human health, holistic health. And this is just a really positive thing that we can do for our soils and for ourselves. Healthy soils equals healthy plants equals healthy people. So I'm happy to share this. Where I'm coming from is Uxbridge, Ontario. I actually went to school at Western in London, Ontario, and it's a beautifully treed city and a beautiful place. Uxbridge is also a beautiful place. There's lots of trees here. As you can see in this picture, I'm surrounded by a mixed deciduous and some softwood trees. And when I first moved here in 2016, my husband and I were hoping to homestead, but we found that our plants would just shrivel up and die. So my garden was really failing. And I began to research ways to grow plants in sandy soil that didn't involve dousing them with fertilizers. So I came across Dr. Elaine Ingham's Soil Foodweb School. And that school has taught me to make really high quality compost. And to stretch the use of that compost a little further, I can turn it into a liquid. So the liquid biological amendment that can be sprayed out on my vegetable gardens. And I've been helping others use it for agricultural purposes and in the arboriculture industry. But it all started with this piece of land with very sandy soil. So a lot of my teaching is coming from Dr. Lane Ingham's Soil Foodweb School. It's a really fantastic program, and I've gone a little further and taken it to become a certified laboratory technician. So I look at compost and soil and compost teas and extracts under a microscope to count the number of microbes. So I can actually do a quantitative analysis of what life is in the soil. And I look for diversity and the right numbers to determine if a compost is truly good for making a compost tea, or if a compost tea is actually safe for application on food crops. So that was all coming from Dr. Lane Ingham, and her main method for making compost, which is a thermal aerobic method. This is just a picture that kind of shows what that would look like. We need lots of aeration. I constantly monitor the temperature with a probe. I'm using clean, diverse feedstock. And even though this can be quite a laborious process, you can adapt these techniques to make a really good compost yourselves. Which is really the first soil restoration tool, is bringing organic matter back into the soil. And you can do that by recycling your waste and turning it into a high quality compost. So you can read more about Elaine Ingham's Soil Foodweb School on that website. And there's lots of YouTube videos out there, and she's well known if you don't know her yet. Okay, so that was my introduction. So now we're going to talk about the soil food web. So in case you haven't had a chance to look under the microscope at 400 times magnification and seen what fungal hyphae, nematodes, and protists and bacteria look like, I'm going to show you. So that's going to really shed some light on the microbiology world under our feet. And I'm going to explain how biology helps restore soil and make better plants. So then I'm going to talk about how to restore soils with biology. So you can use mycorrhizal inoculants. These are products that you can find on the shelf at stores, and they're things like MyPro, Root Rescue. So these are mycorrhizal products, and they're shelf stable, and they're pretty good if you're working with trees. But my favorite stuff is the do-it-yourself compost, compost teas, and extracts. And this is because these products are biodiverse. They can't necessarily grow, like we can't grow all of the different mycorrhizal spores on a petri dish and dry it and put it on a shelf. We can only do that with a small number of the amazing diversity that's out there. But when you're using compost, you're getting hundreds of species of fungi, thousands of species of bacteria, the whole functioning soil food web of nematodes and protists that you just can't put in a jar and put on a shelf because it's a living, breathing organism. And then I'm going to talk about case studies and provide some evidence, and hope to promote this practice and make it more accessible for everybody. And then we'll end with the question period. So starting at the beginning, what is soil? Soil is composed of about 46% minerals, and that means your parent material, so the bedrock that the soil was eroded from. And it could have different chemical properties, textural properties, but typically it's defined by the size of the grains as sand, silt, or clay. Half of that soil profile should be divided between air and water. So there's about 50% pore spaces, and about half of that should be filled with air and half of that should be filled with water. And then 4%, about maybe 5, 6, 7% of that should be organic matter. So like in a market garden where we want to produce lots of vegetables on a intense schedule every year, we want lots of organic matter for them to microbes to feed off of to fuel that high demand environment. Organic matter is an essential part of the soil, and that is where all of the soil life lives. So organic matter is something we're actually lacking in most urban soils. It gets degraded easily. It can be eroded away. It can be scraped away, and it's generally just abused. So we're seeing any soils with below 4% organic matter are struggling to produce healthy plants. So we got to bring that level up to 4 or 5%. So when we send a soil sample in for analysis, like say we want to know how healthy is our soil, a typical lab will send back a report that gives us chemical and physical properties of the soil. Maybe there's a little bit of mention of biology, like the respiration rate, how much carbon dioxide is being emitted by microbes as an indicator of how much life is in the soil, but it's generally neglected because it's complicated to measure the biology, especially the microscopic biology. But the physical stuff we're very familiar with. When you take a look at a soil, you can kind of guess, is it sandy or is it clay? Or is it that nice ideal loam? So that's a very important thing for farmers and growers. Then porosity, that's how much airspace is in the soil. And that airspace allows the soil to hold water or not, or hold air or not. So when we don't have air in the soil, it's called anaerobic, and that can fester pathogenic microbes and be a bad situation for plant roots. And then there's also the issue of compaction. In many urban soils, our soil is so compacted that we couldn't even get a compost tea or a fertilizer into the ground. It would just, or water for that matter, into the ground. It'll just run off. So compaction is a really big physical issue. The important chemical properties mainly are nutrients. So what nutrients are available? And we're looking at the whole suite of macro to micronutrients. So you're more familiar with nitrogen, phosphorus, and potassium as the macronutrients, but there's about 16 other essential plant nutrients that we don't really get in our typical chemical fertilizers. pH is of course important for different reasons, and cation exchange capacity, or CEC, which is the ability of a certain soil to hold on to nutrients. Sand has low CEC, which means the nutrients just wash away, which is why my soil in Uxbridge doesn't grow plants very well. But biology can actually impact the physical and chemical properties of the soil. So we've got macro versus microbiology. People, things that you can see are macro. So people are more familiar with earthworms, centipedes, millipedes, various grubs, even voles and creatures that are digging in the soil profile are your macroscopic life. And then your microscopic life, which I will be showing you lots of pictures of in a moment. So the microbes actually influence the physical properties of the soil. For example, bacteria produce glues that bind particles together. So the sand, silt, and clay particles together in microaggregates. And then fungi or fungal hypha join microaggregates together to make macroaggregates. And then now you've got a nice textured soil. It's kind of sticky, it has air spaces, and it holds water better. So you can take a crappy soil and bring life back to it, and it'll behave more like a loam. Biology actually can influence pH of the soil just around the root zone. They have a buffering capacity. So that rhizosphere, that root zone, will send out, the plant will send out carbohydrates and sugars to attract certain microbes to its root zone and build a symbiotic relationship with those microbes. And those microbes will provide various functions, including pH buffering. They will also make nutrients available to plants. That whole range of nutrients, from the macro to the micronutrients, actually exists in soil. It just is in a form that's unavailable to plants, bound in the sand, silt, and clay particles. But bacteria and fungi chip away at the sand, silt, and clay, and then are consumed by higher levels of the soil food web, who excrete and in that process, turn an insoluble phosphorus to a soluble phosphorus that can be used by the plant. And again, that plant will have a symbiotic relationship with that specific community of microbes, and will cater its exudates, or the carbohydrates and sugars, to attract the right microbes to bring it the right nutrients at the right time. When you use a chemical fertilizer, the plant can't really control how much of that, ionic salt that it gets. It's just there in that dose, or maybe in a slow release. But when you have soil life, the plant won't put out the exudates when it doesn't need the extra nutrients. So as you can see from this holistic diagram, soil health is really a component of physical, chemical, and biological properties of soil. And we need to take all of those things into consideration when we want to grow good plants. So this image is from the Soil Food Web School, which is really the backbone of our talk today. So the Soil Food Web is a web, just like we get in a terrestrial ecosystem, of interacting, consuming along a chain, but it can be way more complicated than a chain. But basically, it starts with the photosynthesizers, so the plants. And they get their energy from the sun, and they put some of that out there to attract the microbes. And when they die, or the leaves fall, or the roots slough off cells, it becomes organic matter. And what breaks down that organic matter? Bacteria and fungi. So they're the primary consumers. And they're having that exchange with the plant roots, delivering nutrients, buffering the pH around the root zone, even providing access to water through mycorrhizal fungi. The next level is the protozoa and the nematodes that eat fungi and bacteria, as well as some arthropods. So micro arthropods are things like mites and springtails that will eat fungi and bacteria. And when they die or slough off cells or produce wastes, the nutrients that were locked up in the bacteria and fungi become plant available. And then moving further, you can get predatory nematodes. And there's a misconception about nematodes that they're a bad thing. In the agricultural industry, when you have like monocultures and then you have a root feeding nematode coming in and wiping out your crop, that root feeding nematode is a big issue. But in a natural system, a couple of root feeding nematodes are natural. But when you have a balance of the different types of nematodes, they will keep that root feeding population in check. That's what the predatory nematodes do. They prefer to feed on the pest nematodes. So a healthy soil food web is going to keep pests in balance. And then, of course, we get into the 4th and 5th trophic levels of higher predators, which could bring us up into the terrestrial realm and just connect everything. So you're familiar with some macroscopic soil organisms like worms, mites, insects, even rodents. These are the main microscopic groups: bacteria, fungi, nematodes, amoeba, flagellates, and ciliates, which are all protozoa, single-celled organisms, rotifers, and springtails. So we're getting to the secondary consumers with those, and those are microarthropods. The images that you see are all taken by my microscope camera at 400 times magnification. I see this kind of stuff all the time, especially in compost samples. Most urban soil samples, all I see is bacteria, so they're not very exciting. And it shows you how different an urban soil is than a forest ecosystem soil. So compost is a lot like a forest ecosystem because there's a lot of decomposition happening in a natural forest with leaf litter. So in a forest ecosystem, we want to see a lot of fungi. That top left image is a fungal hypha. It's dark in color and it's fairly wide. It has nicely spaced cross walls or septa. So that's a beneficial fungi. And I would enumerate that in one of my microbial reports. Top right is the head of a nematode. They're quite large, so I can't capture the whole thing in 400 times magnification, but I focus on the mouth part to figure out if it's a root feeding nematode or a predatory nematode or a bacterial feeder or a fungal feeder, or maybe an omnivore. So in this case, when it has frilly lips, we call it, the mouth part, it has these like little appendages. That's typical of a bacterial feeder, but its mouth is quite large and wide, which is more typical of a predator, or in this case, what it actually is an omnivore. So it can eat bacteria and protozoa. So that's a very beneficial nematode. Bottom left, you can see there's a microaggregate that's being held together by bacteria. There's a couple other things happening in there. Maybe there's a particle of sand, silt, or clay, so a mineral particle. Maybe there's some actinobacteria, those filaments, those are all beneficial. And then there's a testate amoeba. So mostly in soil, we see amoeba living in that test structure with the mouth or an opening at one end where they reach out their pseudopods and collect bacteria to eat. So those are very beneficial. And then the bottom right photo is right in the middle is a protozoa called a flagellate. And you can see one flagella coming off of behind it. And it's hard to identify microbes by photos. Really, we want to use a microscope so we can zoom in and out, or at least use videos. And if this was a video, you would see a flagellate bumbling around. So they're not good swimmers because they only have one flagella. So they just kind of do this. Whereas a ciliate, which is not shown here, but a ciliate is also a protozoa. and it's a really good swimmer. So it has a lot of flagella and it can really dictate where it goes. And they're basically aquatic that way. I mean, all of these microbes are aquatic. We need 50% moisture in our compost pile to keep them happy. But the ciliate is indicative of anaerobic conditions. So 1 or 2 ciliates in a sample is not a big deal, but if we looked at a compost tea and it was full of ciliates, we would know that there's not enough oxygen in there. And when there's not enough oxygen, we can breed pathogenic bacteria like E. coli and Salmonella. So ciliates are indicators that things might have been too wet or too compacted, and that's a compost or a compost tea or soil that is not healthy. So benefits of biology. Did you know that 1 gram or a teaspoon of healthy soil can contain up to 1 billion microorganisms, including several meters of fungal hyphae? That's a lot in 1 teaspoon. So that's healthy soil. And when you realize how much life is in there, you start to get an appreciation of how it plays a role in our soil. So a complete soil food web containing all of those organisms listed on the last page will provide several functions for the soil. I've touched upon most of them. So they provide nutrients to the plant when the plant wants it, the right nutrient, exactly in the root zone. And they can hold on to those nutrients so that they're not simply washed away, and collecting them and then bringing them to the plant, unlike a fertilizer, a chemical fertilizer, which might leach away. They contribute to soil aggregation, so they build micropores and macro aggregates and macro aggregates. Microbes can control pest populations. I mentioned how the predatory nematode prefers to feed on that detrimental root feeding nematode, but certain fungi will also wrap themselves around plant roots, protecting that plant root from being penetrated by a detrimental fungi. And bacteria will do this too. That's more of an indirect way to prevent a pest from coming in, but then there's the actual predatory effect as well. So in different ways. You may have heard of certain fungi or mushrooms being used for mycoremediation. So where there's been mining sites, spill sites, polluted sites, scientists are bringing in certain species of fungi to break down those toxins, and they're very good at it. And as I mentioned too, biology can buffer pH, so a plant can better survive in different types of soil. So I'm going to focus now on mycorrhizae, because this is a very special and important type of fungi for trees. Myco means mushroom, rhizae means root. And 80% of all plants form a symbiotic connection with mycorrhizal fungi. So they need 80% of all plants. Mycorrhizal fungi will attach themselves to the root hair and move outwards, as this photo on the right shows. The tree on the right is inoculated with mycorrhizal fungi. And you can just imagine how much more surface area those roots that plant has access to. The mycelium can penetrate tight spaces and reach up to 50 times the length of a tree that does not have mycorrhizal inoculation, increasing the surface area hundreds of times, allowing that plant to access nutrients and water more easily through the mycorrhizae. Mycorrhizae also break down organic matter to release nutrients. They can inhibit the root-feeding nematodes and other soil-borne pests by coating the whole root. It improves the tolerance to stress. So you can imagine that a tree with that much more access to water is going to do better in drought conditions. And one study shows that inoculating soil with mycorrhizal fungi can increase plant yields by up to 40%. And there's more than just this one study, but I'm just highlighting that. So if you were to purchase mycorrhizal inoculant, you need to know the difference between the 2 major types of mycorrhizae, endomycorrhizae and ectomycorrhizae. So endo, they're way more common. They're found in about 85% of plant species, or sorry, out of the 80% of plants that need mycorrhizae, it makes 85%. And they're found in many flowering, leafy fruiting plants. So the stuff that you buy for gardening, attractive plants, they're going to need the endomycorrhizae. And they actually penetrate the root cell and form a tight symbiosis by entering the cell. Ectomycorrhizae are found in mostly just conifers and hardwoods. In conifers, they're often obligate symbionts. That means that they're symbiotic and it's obligatory. So that tree, that conifer, will not survive without an ectomycorrhizae. And they figured this out in, I believe, the 1970s when they transplanted trees from one place to another place far away, and those trees all died until they brought in some native soil that contained the ectomycorrhizae, and it was the only way that that conifer would survive. Biologically or visually, I should say, the hyphae do not penetrate the root cells. You may have seen the movie Fantastic Fungi, and that's an exciting view that I wanted to share. So this is an image from that. If you were to delete all of the, soil particles, delete most of them so that you could actually see the fungal hypha, it would look something like this. So you have a tree root there, and you can see the network of mycelium or fungal hypha. When you have many hypha, it's called mycelium. And the hypha here on the right is touching a soil particle. So an aggregate of some kind, like a little rock, it could be a little piece of sand, we'll say. And it's chipping away using glues and enzymes, chipping away at that particle to extract phosphorus and other nutrients, which it then passes through its network to reach the tree root. Here's another still. So imagining that the soil is deleted, and this is what you would see. You have two trees here, and you have an extension of mycelium. And there's even recent evidence that suggests that trees and other plants will share resources using mycelial networks. So just like the tree philosophy of right tree, right place, we have the same thing with right microbes, right place. So when I'm looking at my samples under the microscope and I'm counting bacteria and I'm counting fungi and I'm enumerating them in micrograms per grams, I can create a ratio of fungi to bacteria. A higher fungal to bacterial ratio means there's more fungi than bacteria, and vice versa. So when you consider an ecosystem that has been damaged, like say a forest fire or tillage, the first thing that pops up are weeds. And those weeds have created a relationship, they have evolved, co-evolved with the first microbe that comes up, bacteria. Bacteria reproduce really fast, and they're the first thing to start to prime the ecosystem for succession. So weeds and bacteria aren't necessarily a bad thing, but they do indicate disturbed soil. And if you want to grow early grasses and brassicas, you want a very low fungal to bacterial ratio, which means a bacterial dominant compost or compost tea. It's really easy to make a bacterial dominant compost. It's just composted manure. Bacteria like to feed off of high nitrogen manure. So most of the compost that's in the marketplace is actually pretty much just bacteria, which means you're pretty much just feeding the lower successional plants. If you want to feed the soil for anything that's woody or higher successional, you need to increase your fungal component. So looking at shrubs, vines, and bushes, you have more fungi than bacteria. And then moving into old growth forest, you have very high fungal to bacterial ratios. So you can use your compost or your compost tea to address those certain plant successional stages. So restoring soil with biology. I don't like to push some kind of instant solution or band-aid solution. I think the only way that we're really going to restore soil is through a multidisciplinary approach or a holistic approach, something that involves people, plants, animals, the environment, what's going to actually be sustainable. So I like to borrow some ideas from permaculture. So earth care, people care, fair shares to, keep it going. So permaculture is an approach to land management and settlement design that adopts arrangements observed in flourishing natural systems. And I truly believe that if we want to really restore the soil, we need to work with nature instead of creating band-aid solutions. I also love the concept of regenerative agriculture. So all of these things in this graph, like keeping soil covered, maintaining living root year round, integrating livestock, so diversity, and minimizing soil disturbance are all going to restore biology to your soil too. All right, let's get into amendments and what you can do. So I've broken it down into the traditional types of amendments and then the biological amendments. And then I've further broken it down into solid and liquid just because it falls easily that way. So solid amendments, your first go-to really should be compost and mulch, especially for trees. You have to mulch trees. Compost and mulch provide organic matter. They actually have microbes in them, so that becomes a biological amendment. But just even in their physical properties, they're going to help maintain more water in the root zone of your plant, and the mulch is going to keep the water there, keep the roots cooler, and keep weeds from getting in there. Organic fertilizers can work too. There's a whole range of organic fertilizers, and just some examples are blood meal, which is very high in nitrogen, rock dust and biochar, which can be integrated with the soil to enhance the habitat for microbes. Like biochar and rock dust provide apartment buildings for microbes to live and flourish, and adds micropores and airspace. Liquid amendments, so again, organic fertilizers. Some examples are liquid kelp extract and fish hydrolysate. Personally, I never want to work with fish hydrolysate again. I don't mean to turn anybody off of it, but it's oily and the smell won't leave you for a week. So kelp extract, it's kind of expensive, but it's a good amendment. And there's so many more. So biologicals, solid biological amendments, again, compost and mulch. As I mentioned, they have the microbial properties. Mycorrhizal inoculants, which I guess are solid, but you mix your spores or propagules in water, so it becomes a liquid, and then you can inject it into the root zone or dump it around the root zone while transplanting. And there's a number of beneficial insects, and some of them are microscopic, that can be used in the biological amendment category. And then the liquid biological amendments are compost teas and extracts, protozoan infusions, and nematode extractions. And I'll get into the teas and extracts because that's my thing. Okay, so compost. Compost is also my thing because it all starts with compost. That's the inoculum. So compost is more than just organic matter, and it's definitely not organic wastes. So everyone can sell compost. It's just technically degrading biological material, but it comes in such a huge range of qualities. Municipal compost is super questionable. I know it's treated in a certain way, but when you look at the starting feedstock and you know that people are throwing in a lot of microplastics, you might not want to use that for your edible crops or your compost teas. You want to know what you're working with. So a really good compost should be as the thermal aerobic method by Dr. Lane Ingham that I mentioned, so that the heat kills the weed seeds, and the turning or adding oxygen to the pile promotes the growth of aerobic beneficial microorganisms. And ideally, compost suppliers should be providing people with information about their compost, like traditional soil tests and microbial analysis. If you're looking to use that compost for compost tea or extract, you really want that microbial analysis. And I've looked at many commercial suppliers of compost, and I can say that they do mostly contain just bacteria, and they lack the whole soil food web. And that depends too on how it's stored. If it's just stored baking in the sun, the UV is going to hurt the microbes. There's not enough turning, there's just not attention to detail. And it's hard to do. It's really hard to get enough good compost for the restoration jobs that we need. So I would love to see more people trying to do good composting. And I'm working on a database for high quality compost for people to purchase. So do-it-yourself compost. Here's how you do it. There's a great instructional video by one of my other mentors, Matt Powers, the permaculture student. So check it out, Google it. But essentially, here's how it works. The first image shows diverse feedstock, and you need to balance the amount of high nitrogen manure or beer mash or legumes All of those are high nitrogen, and bacteria love that. And that's an important part of getting reproduction going among the bacteria, because that's what drives the heat up. And then in this second image, you can see you want that heat to be in that red zone. So you want hot compost, so hot that it will kill the weed seeds. Then you need to turn it after 3 days so that the weed seeds are cooked. You need to turn it, reintroducing oxygen to the pile to make sure it doesn't go anaerobic, but also to get all of the parts that were on the outside of the pile to the new middle of the pile so that that gets cooked. And you want to turn it 3 times within about 15 days while it's still hot to cook the whole compost and keep it aerobic. And then it is technically safe to use. But then you want to age it, and aging it is going to allow the fungal properties, fungi to increase because they reproduce much slower. All right, so let's talk about mycorrhizal inoculants. As I mentioned, there's the 2 main types, and just a refresher, the endo, also called arbuscular mycorrhizal fungi, are best for leafy, green, fruiting, flowering plants. And about 80% of the plants that need mycorrhizal fungi are going to need the endo type. But the trees are going to need ectomycorrhizal fungi. 10% of all plant species, but most trees and most conifers. These are sold as spores. And unfortunately, the spores aren't always viable. And it's found that the ectomycorrhizal products are variable in how effective they are and how efficient they are. So the drawbacks are when you buy an inoculant that is lab grown, it's going to be less diverse than, say, a compost. And it might not contain the right species to make that attraction with that plant, that symbiotic relationship. It's not necessarily viable, as I mentioned, and there's need for better quality control. So this is an emerging market still, and it can be expensive. Here's an example. I think Root Rescue is a good product because it actually contains 18 species, including endo and ectomycorrhizae. It's simple to use. I used it a lot when I was an arborist, and we were doing root zone rejuvenation of boulevard trees. So we used an aerator to blow all the soil away from the root zone, and then we filled it with compost, some of Root Rescue mixed in with water, and poured that into the root zone, and then topped it up with mulch. But this is what I love. This is the fun boutique do-it-yourself stuff. And you can try to do it yourself, and I encourage it. I can also make it for you, and I can also sell you kits so that you can more easily make it yourself. So what is compost tea and extract? They're both considered liquid biological amendments. And when I tried to take this to the Canadian Food Inspection Agency to get it certified for use on food, they called it a liquid consortium. Which makes sense because it's a consortium of microbes. They're all different. Unfortunately, the CFIA isn't really aware or ready to accept compost tea to approve it for use on food because we can't standardize it. It can't have a guaranteed analysis like a chemical fertilizer can. And it actually defeats the purpose if we were to try to keep our compost tea consistent, because we want to make different brews or different fungal to bacterial ratios for different types of plants. And diversity is key. And these are microbes. They're a living, breathing community. So they're active and they're reproducing, and that community is changing by the minute. Bacteria reproduce 4 to 20 times their population size in, I think, 20 minutes or 10 minutes, a lot. But yeah, compost tea is ready to work. It needs to get put out in the soil as soon as it's made. And I see it as a restoration tool, but not a miracle cure. So keeping in mind that we need to first address issues like compaction and the use of chemicals that have high salt concentrations that make an inhospitable environment for those microbes. We need to cut that back first. We need to add a compost tea. We need to add compost, mulch, whatever it is that particular site needs. So yeah, compost tea is not a miracle cure. Drawbacks, it has limited shelf life. So you can't actually seal it in a jar for very long because it needs oxygen. And because the bacteria are reproducing and they're using up oxygen, we're we have the potential to quickly become anaerobic and produce, if there was any E. coli in there, it could reproduce. So best to use it as soon as it comes out of the brewer and it has the best diversity and aeration. But maximum for compost tea, 24 hours. If you put it in the fridge, you can prolong that just maybe another 12 hours. But remember, the community is changing and it's just best fresh. As I mentioned, it has a variable community composition and it's difficult to standardize. So, you know, one guy can sell compost tea, another person can sell compost tea, and they're totally different and people don't really know what they're getting. So it should be quality controlled by a microscope and supported with in-field tests. Not a lot of people know how to use the microscope and actively count their microbes, but it would be great if people who were selling these products did. One thing I wanted to say is we want to avoid selling snake oil and we want to sell a good product to build credibility for compost tea. And that's one of the challenges of getting it into the marketplace, is that there has been a lot of misinformation and lack of quality control. So the difference between compost extract and compost tea, well, actually the similarity is that they both start with a high quality compost and non-chlorinated water. Then you take a fine mesh bag, you put your compost in, and for the compost extract, you massage it by hand. The microbes are extracted out into the solution, and that's your compost extract. But you might want to do that a few times to get the numbers to where you want. So it requires more compost. It can actually be preserved a little bit longer, so maybe 48 hours, like 2 to 4 days max, and is best for use in soil drench, and I'll explain why. Compost tea, it contains the whole soil food web, but because we have added microbial foods, the bacteria are reproducing the fastest, so the community composition becomes more bacterial dominant, so it decreases the fungal to bacterial ratio, which depending on the plant successional stage that we're using, we might not have as much value for. But on the plus side, it requires less high quality compost. And since high quality compost is so hard to come by, sometimes we need to actually use a compost tea to get our numbers up really high. So you don't have to repeat that extraction process. You can just put the compost in the bag in a brewer that is aerated, add the microbial foods, and let that go for 24 hours. And yeah, so here's some caveats. It comes out somewhat sticky, like very little, not really noticeable, but because those bacteria are reproducing, they're producing a biofilm, and that's getting on the inside of your brewer and in your piping. You're going to have to clean that. But because it's sticky, it makes for a good foliar spray. So if you're treating leaves, compost tea is good. But because the bacteria are reproducing and using up oxygen, and if we don't have enough good aeration, we could start to breed anaerobic pathogens. So we want to be more careful with this product. We want to use it within 24 hours. And then the compost extract, again, I said best for soil drench because there's no added microbial foods. So there's no reproduction happening. There's no glues being produced. So it's going to soak into the soil better. Both of these products you can deep root inject into the root zone to help it get down. So there's a range of brewers. A lot of these are do-it-yourself style brewers, or if they're not, they're very expensive. So they range in size from 5 gallons to 250 gallons, which is a 1,000-liter tote, and way more than that in agricultural settings. That's just what I've worked with. So it's just to recap, you need high quality compost, non-chlorinated water, actively aerate the solution. So you want to see a rolling bubble. You need microbial foods added, so you can work with different recipes to promote, get the right microbes, get good concentrations of diverse microbes. You can add other enhancements just before application, like yucca extract. You can add more stuff to send your compost tea out onto a field to allow the microbes to grow more in the field. And yeah, there's a whole bunch of different recipes. So then you brew for 24 hours and apply within 24 hours. And there, compost extract, simply put compost in water, agitate to extract microbes into the solution, repeat with more compost and apply. I really want to make compost tea more accessible to people. So I'm making a high quality compost and testing it under the microscope and testing different recipes. So I'm developing a few different recipes and I'm selling a kit. It's a 5-gallon bucket kit. It will come with an instructional video, the airlift brewer style. So that manifold is called an airlift brewer. It was designed by a man named Tim Wilson, and we have permission from Tim Wilson to manufacture this brewer. It is tested by Lane Ingham Soil Foodweb School, and it is considered the best on the market for getting a good aeration and producing a good diverse microbial community. So if anyone is interested in that, reach out to me, and I want to help people make compost tea. How do you apply it? You can simply put it in a watering can like I did there. Here I was working with my arborist friend in my former place of work, and we're deep root injecting compost tea for all of these boulevard trees, which really weren't doing too well. And I think these trees needed a little bit more love than compost tea, to be honest. So the application rate is 25 gallons per acre. So you can imagine a 5-gallon bucket, that's 0.2 acres. That's a lot. The way that you would spread it out is by adding water. So you can dilute it up to 20 parts water. The equipment for industries or commercial businesses that are doing, like landscaping companies, arborist companies that are doing foliar sprays, surface drench, deep root injections, they have equipment for that. They're going to use low pressure PSI so that they don't squash the microbes as it passes through the hoses and spray guns and stuff. But at home, you can use a watering can or maybe a backpack sprayer. And if you're in the agriculture business, you can actually put the compost tea diluted to 20 parts with water in a tractor and spray it out or drip or irrigate it out that way. And that's the best way of covering a lot of land, because as you can see, if 25 gallons covers an acre and you've got a 100-gallon brewer, you're covering acreage. And then very important that everyone always forgets is to clean your equipment. Yeah, so like industries, commercial businesses are on the go, but they have to set this time aside to wipe the surfaces, the interior surfaces of all the brew equipment. We just want to make sure that all the biofilm is washed out so that there's no potentially anaerobic pathogens stuck in there that will contaminate future brews. So we clean every time. All right, we're getting close to the end of the presentation, so I'm just going to wrap up here. We've got some proof that compost tea works. These are 2 little experiments that I did at home, but we really need more studies. I want to see more scientific studies at the university level, but the question is, who is funding the research? A lot of this is do it yourself at home, so we don't have the big ag companies funding this research. There's also issues with questionable products, lack of quality control, variable results, and limited testing, because we need a microscope, we need skills with the microscope, we need some kind of standardization, which we do have, and I'll get to in a second. On the Soil Foodweb School webpage, there are a few case studies that you can look at where we've seen excellent results with compost tea. In this study by Renald Floor in a market garden, he used different scenarios: a control where nothing was done, a compost only, a microbial inoculant with no compost, and a microbial inoculant with compost. So like he used a protozoan infusion. With the inoculation, he saw 38% less weed biomass than in the control. And then with the compost as well, also a reduction. But most interesting was his yield results, where all 8 crops that he grew increased biomass by, on average, 72%. So I said about proof and counting microbes and getting quantitative data. The Soil Foodweb School has that certification program. I've taken it, and I can produce these kinds of reports where I actually get the fungal to bacterial ratio and I detect any pathogens. It's not as detailed as a DNA analysis, but it is cheaper than a DNA analysis. And it's a great tool for before and after studies to see if a program worked. or to analyze different composts and pick the right compost. So this is what the data output looks like. I formatted it in different ways, but here I compared my compost, an extract, and a tea. It was just a little study I did to see if my qualities met the minimum standards by the Soil Food Web School, which they did, which was really great. My next project is to do this kind of testing on compost throughout the GTA, so I can build a database or a website of quality suppliers so people can get biological compost, which, by the way, is going to cost a lot more, but it's worth it. So if you need anything like compost, teas or extracts, consulting advice, or microbial assessments, reach out to me. My number and email are on this page. Also give me a follow on Instagram. I'm actually Rewild Uxbridge, so it's kind of like Reforest London. And I love to share what I'm doing and, give advice and have fun. I host events too that are really fun. So there's my reference page, and I'm ready for questions.
Thanks so much, Colleen. That was super in depth, and I think that I have a couple questions, but I'll open it up to the chat. If anybody has any questions right off the bat, please feel free to put those in there. Yeah, for Colleen. I guess I can ask my question. I don't know a whole lot about foliar treatments. Can you talk about how that helps plants? What is spraying like that? Like, what does it do?
Yeah. Okay. So when there's a nutrient deficiency in a tree or other plant, maybe you can detect that from the coloration, or maybe you've done a foliar test. Plants actually pick up nutrients a lot faster through their leaves than they do through their roots. So that's a really quick fix. It works really well with iron deficient plants. But it works in a similar way with the microbes too. All surfaces on your body, on any living thing, are covered with microbes. So when we put the beneficial microbes on the plant leaf, we can actually fight plant pests like sooty mold, that competition theory where beneficials outcompete the detrimentals. So there's benefits of putting compost tea on any leaf surface that has a nutrient deficiency, because there's also nutrients in compost tea, or pest issue.
Okay, great. Yeah, thank you. A question has popped up in the chat. What are your thoughts about alfalfa pellets as a compost?
I love it. I've been getting more into what they call vegan composting, so we're not using manure as much, and it's safer because manure contains salmonella and other pathogens. Alfalfa pellets and beer mash is what I tend to use because I have some, are high nitrogen sources that don't contain any pathogens, and they get your compost really hot. So when you get the balance right and you use the alfalfa or beer mash in the right ratio, you get a successful thermal aerobic compost without having to use manures. So go for it.
Okay, cool. Thank you. Yeah, anyone else, if you have any questions, put those in the chat for Colleen. What is beer mash? Is a follow-up.
It's a byproduct of the beer brewing industry. And I'll be honest, it's like super hard to come by because in recent years, everyone's onto it. So farmers actually feed their livestock the beer mash. It's like fermented, and in some ways it's good for them, and in some ways it's kind of just a filler for them. But all of my compost friends are just fighting over it because it's a high nitrogen spent grain. It's wet, so it's ready to go into the compost pile, and it smells good, and there's like no pathogens, but you need to use it right away. Yeah, so it's a really great composting feedstock.
Cool. Yeah, I've definitely heard of like the spent grain. I've maybe seen that kind of uptake in the recent years. There's another question here. Is there such thing as a home compost, in brackets, clean kitchen scraps and autumn leaves only, that can be detrimental to a veggie, herb, or fruit garden? I guess my question is, could I be harming versus helping?
It really depends on how that compost is managed. Like if it's really wet, it could be detrimental. We want to have about 60% leaf material or brown material that can soak up the extra water, but also provide enough carbon to like balance the bacterial to fungal ratio. So yeah, that's that's kind of a tricky question. If you want to send me an email though and give me more information about how you're building that compost, I can help you make sure that it is safe for your food. Yeah, I'm hoping that it is.
Great, yeah. Thanks. I hope so too. Two more questions here. Thoughts about worm castings that you can buy from a store versus homegrown?
I still think homegrown is the best, which is like why with the last question, I would say like it's probably good because of the issues with storage. So like vermi castings that are old and dried out and put in a bag that can't breathe. Like there's like vermi castings are a really great inoculum, and they contain mostly beneficial, if not only beneficial microbes, because as food passes through the gut of the worm, they somehow get rid of the pathogens. So vermicompost is great, but it's probably best to make it fresh. Do it yourself.
Great, yeah. Another question here for you. What happens if compost turns anaerobic?
Um, it starts to like produce alcohols that burn plant roots. So I think it becomes acidic. If you were to use that anaerobic compost, it would obviously be pretty bad that way. But it could also contain anaerobic bacteria, which are Salmonella, E. coli, and other pathogens, and breed them. So that's not going to help you or your soil that much. Although there is a whole field of successfully doing anaerobic compost, like bokashi and stuff from Korean natural farming, KNF. And I haven't gotten into it, but there's a way to do it, and there is a benefit to anaerobic bacteria and stuff. So just be really careful. What I would do though, if my pile went anaerobic, is I'd spread it out, let it dry up, and then add a bunch more brown material and restart it.
Oh, so you can still work that back in.
Yeah. All of the waste is recyclable.
Mhm. That's great. Um, I have a couple more questions. Oh, one more question here. Would beer mash need to be put through the compost first, or can it be used directly in place of manure?
It can be used in place of manure, yeah. When you start a thermal aerobic compost, everything has to start all at once. So there's a bunch of caveats and it's tricky. And I recommend you watch that video from Matt Powers or any video online with a bit of research. Because it's tricky. Thermal aerobic compost has to happen within a short time frame. You need to monitor it. Like you can monitor it with your nose a little bit and your hand, make sure it's wet. And if it smells bad, it's probably gone anaerobic. Yeah. Sorry, I got sidetracked. I lost the
No,
question.
totally. Yeah, so I know that there's like different manures need to be composted prior to going in your garden.
Oh,
Um,
oh, that's maybe what that means. No, beer mash is considered sterile because it's gone through a high heat, which has killed all of the life.
so it can just be worked right in.
Yeah, you can
Um,
actually even, if you're not able to maintain your heat in your compost pile for 3 turns, you can add fresh beer mash and
mm,
it's not considered contamination. It can boost the temperature and keep it going through the thermal stage.
cool, very cool. I think we'll take this as a last question and then we'll let you go. For regenerative gardening, do you recommend a winter crop over another? I'm planning to use oats as they die on their own and is less work.
I'll be honest, I don't know. I would love to learn more about regenerative agriculture. And I'm getting into it with some farmer friends, but it's not my area of expertise. So yeah, I'm not going to try to
Yeah,
answer that
fair.
one.
All
Sorry.
good. I will say I did hear, I don't know if this is because of the mild winter, but I heard that some people that were using oats as a cover crop that they expected them to die and then spring came along and they hadn't died off. So just an interesting tidbit about maybe our changing environment as well. But
Yeah. And
yeah.
I do want to wrap up by saying something is working with nature and with biology is way more dynamic and variable and challenging than working with our band-aid solutions of chemicals in the past. So don't be discouraged if it takes like 3 years to figure out how to grow something. I've been doing this for I think 7 years now, and I'm finally like fine-tuning things with the compost. But yeah, I don't know everything, and And yeah, it's going to take hard work, but this is where the future has to go to have a healthy environment and healthy people.
Absolutely. Yeah. Well, thank you so much for being here today. It's important for us as we learn what we can do for our soils and especially in tree planting and apply this in many other ways to hopefully restore that ecosystem that I think is so easily overlooked. So
Yeah,
yeah, thank you so much, Colleen.
my pleasure. Thank you.
All right. Have a great day, everyone. And yeah, we'll see you at next webinar next week. All right. Thanks.
Bye.